Thank you. Thank you for inviting me here. Humbled to be here and give a talk. So it will. The title is not my quote, but it will become apparent why it's the title in a couple of slides. I think it became also apparent from the previous talks that microbes play a lot of role in this planet. You can find them in every corner of this planet, and they're the most ancient form of life here, and they've been shaping the planet through billions of years of evolution. And I think the current numbers, so Knut talked this morning, there are about 10 in the 30 microbial cells in the planet. In terms of bacteria, there are 10 in the 12 different microbial species. So there's quite a bit of diversity estimated currently on Earth, and they colonize every biotic and abiotic surface. And the majority of them, as Knut showed, live in communities. So basically this idea of understanding microbial communities in the ecological settings is this idea of studying microbial ecosystems. And the realization that we live in a planet that is full of bacteria, I would say for the general public, it's not very old. And if you go back to history and understand how people think about microbes, I would say that it had to be till we were able to sequence and understand the diversity that's out there that we understood that. And this is actually a quote from this planet of bacteria. It's a quote by an evolutionary biologist and paleontologist. He's died now, so Stephen Jay Gould, in 1996, in the early times of being able to detect bacterial diversity outside in different parts of the planet, he made this quite influential article in Washington Post saying that what is the evidence that bacteria have been there forever, and the diversity is huge and colonizing every corner of this planet. And you can see that they can colonize from animals, inside or on animals, but they can colonize also every kind of surface of the planet, no matter how hostile it looks, how high the temperature it is, how cold it is, or what the salinity or the desiccation is. And if you look, many of those microbes look morphologically different, but this is not the way to understand their diversity, because a lot of the microbes look exactly like others in terms of morphology, and this has hampered a little bit our ability to understand diversity. And we had to go down to be able to sequence, and through work of seminal work from Carl Vose and then Norman Pace, being able to identify species based on the 16S marker. And then it's when we realize how diverse are things there. So this, not to forget that bacteria have been there in the planet way before there was oxygen in the planet. There's reason that there is oxygen in the planet. That's why also you find them also in space when humans bring them there. And this is microbes growing on the outside of the International Space Station. So how large is this diversity? So this is just to give you an idea about the diversity. The estimate I told you is 1 trillion microbial species in the planet. The red dot here is just a 10-fold magnification of all animal and plant species put together, just to put it out as proportion. And you heard the talk of François earlier this day, that bacterial species are things that evolving also and exchanging genes horizontally. So the entire meaning of pangenome means that two strains of the same species can carry up to 50% of the genomic content different. So this is a different way of thinking about species in the microbial world. So now coming back to the original title of what I have, that the realization that bacteria are everywhere is not new, is actually much older. And this is where the quote coming from. And the quote actually is from Bas Becking. He comes from the Delft School. He came from the Delft School, and he said this in quite influential article in 1934 for environmental microbiologists, that everything is everywhere, so microbes are everywhere, but the environment selects. And the Delft School has been started by Martinus Bayernik back in the early of the 20th century. He did a lot of seminal work understanding about nitrogen fixation, figuring out that microbes can use a lot of different electron acceptors, including sulfate, basically, to respire. But the idea is that microbes are everywhere, so if you take from different parts outside in nature and you put things to grow, there are lots of things growing, but the environment would select what it grow. And you can have things that there are too many things growing, but you can also have selective media that only specific things will grow. And in that sense, so microbial ecosystems are everywhere, and I think we saw, so this was the enrichment culture idea, so microbial ecosystems are everywhere, but it took a few, I would say, reductionist microbiologists and molecular biologists in the 50s and the 60s. We tend to study a single species in isolation and understand molecular mechanisms. So things are changing, and people are starting now to understand communities, and you did see this picture before that just shows how those communities are sometimes in nature. So this is the gut epithelial line the gut epithelial line with the mucus in green here, and these epithelial cells, and those are microbes, and those are fish probes on the two major phyla, basically, of microbes. So microbial communities are diverse and dense in the gut, and for me this is a prelude to tell you why we think that the human gut microbiome is an interesting microbial ecosystem to understand those communities. And so I have a few things here where I'm quoting why the gut microbiome is one of those systems that they're good to start understanding microbial communities. Is obviously the community at the moment that we know the most about. So there's seminal work in the last couple of decades. So some impressive numbers that many of you might have heard before, that you might carry as many microbial cells in your gut as you carry human cells in your body, that they code for 100 times more genes than your human genome. And although you're 99% identical in terms of your genome between two individuals, you're more than 90% different in terms of your microbiome. So those are impressive numbers. That makes microbiome quite interesting. But I think what is more relevant for using it as a microbial ecosystem is that it has key characteristics that a lot of communities have out there. So it's a very diverse community. So it's between 250 and 500 species that you carry in your gut. It's a very stable community, so across all your adult life, you more or less carry the same microbiome and the same strains. So the example of with E. coli being transiently colonizing and getting a different strain is more the outlier rather than the common in the human gut. And they have characteristics that a lot of communities have in closed systems. They give also colonization resistance, so they're very recalcitrant to intruders, and they fight off intruders. The other thing is that there's a lot of technologies that have been developed now that one can go beyond just studying microbiomes just by seeing what is there, but you can actually go and perturb the system in order to understand beyond correlation, causality, and of course it's important for human health. So this is some of the reasons that several people are working on the human gut microbiome, also not only from a human health perspective, but also to understand how microbial communities behave and interact with the environment. So this is a little bit on what you can do nowadays. So apart from observing what's happening, you can actually go and make communities, synthetic communities. So this is all the prevalent and abundant species on the human gut. So you can cultivate many of them. So here they're ranked ordered based on the genus, and you can see the species diversity they have, and so you can see the prevalence here. So for example, at the EMBL and in several labs, we have more than 100 different species that you can grow in the human gut microbiome. You can do genetics on them. On some of them, you can put them together and grow synthetic communities, and you can also phenotype them. And this way you can start asking questions, simple questions about microbial ecology, and you can ask what does make those communities stable, and what does define this interpersonal variation that it is. And people have been doing that at an association level, so you can get large cohorts of thousands of individuals and say, what other parameters are explaining inter-individual difference in terms of microbiome composition? And this is one of the very first papers, came in 2016 by Jeroen Reis's lab in KU Leuven. And you can see in the circle here that you can explain only a very small part of the variation. So you can explain about 16% of the variation that two individuals would have. But the part that you can explain, actually, if you see the first thing that comes is medication. There's also about diet, there's also lifestyle. So there's a lot of things that have to do with the environment that defined what the microbiome is there. And as the cohorts actually are increasing, this is becoming more and more obvious. So the numbers are increases. You can explain more of the variation. By now, you can explain about 25 to 30% of the variation. That's a more recent paper making the big claim that actually the environment dominates over the host genetics in what makes the human gut microbiota. So when we started thinking about environment, we thought that medication is perhaps some of the most bioactive molecules that we as humans consume. And so we want to see what is the direct effect they have on the gut microbiota. And there was quite a bit of literature out there when we started those studies. Maybe the poster child is metformin. This is a type 2 diabetes drug that actually has been shown by now that part of its anti-hyperglycemic effect goes through the microbiome. So the microbiome is part of the therapeutic effect of the drug. So there were multiple papers in the early 2010s making the claim that type 2 diabetes patients have a different microbiota composition, only for people to realize that there was a huge confounding factor in all those studies that was coming from the main drug that people in Western societies get, and that was metformin. So metformin is causing the changes on the microbiome of people that have type 2 diabetes. And then there were people that went and did also metformin that would only very late release in the colon and showed that basically a lot of the anti-hyperglycemic effect is much more effective now than when it late released in the colon. They took also stool from mice and transplanted to mice that have never seen metformin before and could reproduce this anti-hyperglycemic effect. So this is the poster child of a drug that not only affects the microbiome, but the effect on the microbiome has also to do with the therapeutic effect on the host. There are a few other anecdotal studies that proton pump inhibitors in big cohort, people that take them have different microbiotas, and also with NSAIDs and atypical antipsychotics. So when we started back then, what we want to see is that how many of those effects might be direct. Is direct effect of the drug to the microbes, or does the effect go through the therapeutic effect on the host? What is the extent, and if there's any relevance in terms of therapeutic effect, or is it just side effect? So in order to do that, I'll show you three different small stories on this kind of realm, trying to figure out how drugs interact with microbes. The first thing has to do with non-antibiotics, and that's work that Lisa Meyer a few years back did in my lab. So this is published work since quite a few years, so I will go a little bit quick on that, but I just want to set the stage for what comes next. So what we did back then is that we took a lot of drugs that the FDA-approved drugs. Here you see there are 1,200 different drugs. Only a small part of the pie is anti-infective. The rest of the drugs have bona fide targets only in eukaryotic cells and human cells, not in bacterial cells. And what we did is that we took 40 of the most prevalent abundant species in the gut, and we asked directly how many of those drugs directly inhibit those species. So those microbes, you can only, they grow only under anaerobic conditions. So we did the whole screen anaerobically. And what we immediately saw is that the impact of non-antibiotic drugs on the human gut is extensive. So at least a quarter of them inhibited one gut microbe. I'm showing you the data. So on the x-axis, you see 1,200 different drugs. On the y-axis, you see the 40 different species. There are three categories: green is antibiotics, orange is drugs that have human targets, and the purple ones are basically other anti-infectives. That means antifungals, antivirals, and antiparasitic drugs. Every time you see a colored stripe, that means that this particular drug is inhibiting a particular microbe. We can treat the antibiotics as control at the moment, but you see that out of 800 human-targeted drugs, about a quarter of them target at least one microbe. Some of them target many different microbes, almost similarly like antibiotics. So we went further and we looked if it has any in vivo relevance. Actually, we could see for the few drugs that there are clinical data there, we could see concordance between the in vitro data that we would see and what people see in patients. And we can even actually use the side effects those drugs have in humans to predict what other drugs would have basically effect on the human gut microbiota. And we did that and it was working well. So you can see that there are specific therapeutic classes that they hugely enrich on targeting also microbes. Two of them are actually cancer drugs and antimetabolites. The second one is antipsychotics. There's quite active work going on antipsychotics, how much of the effect on the gut microbiota affects actually the therapeutic effect on the patient. And last but not least, we saw that, of course, microbes, if you inhibit the growth, this puts selective pressures for developing resistance against that drug. And the question is that what are the resistant mechanisms for those drugs? And some of those resistant mechanisms are broad resistant mechanisms that you also confer antibiotic resistance. So a line of research in the lab is going about how much common is this resistant mechanisms and whether polypharmacy is driving antibiotic resistance. Now, before I show you the second part about antibiotics, what I want to make a point is that I'm saying that the environment and harsh environments that, like bioactive molecules, are impacting microbes. But I told you from the beginning, microbes can also impact the environment. So you can ask also the question, how much microbes are affecting drugs? And this is work by Michi Zimmermann, who's my colleague of mine at EMBL nowadays. He did that work when he was a postdoc. So they did a similar screen, just instead of testing how much drugs affect microbes, they test how much microbes are affecting drug concentration. This is 76 different microbes, about 300 different drugs. Every time you see more white or more grayish boxes, these drugs are metabolized by those microbes. And we did also work with Kiran Patil, who used to be at EMBL. And we saw that basically not only drugs can be metabolized by microbes, but there are specific microbes that accumulate intracellular drugs without that affecting visually their phenotypes or their fitness. But that affects the availability of the drug for the community. So microbes have also an ability to impact non-antibiotic drug bioavailability. So I show you about human target drugs, what happens on antibiotics, and that's work again that Lisa Meyer started in the lab, and then Camille Goemans is another very talented postdoc in the lab, also continued on that. And the idea was that antibiotics, I told you, well, I have to mention that actually I always have to pay tribute to Martin Blaser when I'm talking about antibiotic effects on the gut microbiota. So Martin has been doing seminal work on that, making the point that antibiotics as collateral damage may be also impacting the gut microbiota, and this change can have impact on many different behaviors on animal models. So what we did is that I showed you this plot before. We had a lot of different antibiotics, so I think in the order of 160 over 40 different species. And I told you as a control, you can say that antibiotics are broad spectrum, so they're targeting pathogens, but they're also targeting commensals, so no surprise. But if you look actually more carefully, you can see that there's a clear separation on drug classes and what happens. So the antibiotics like aminoglycosides that don't work unless microbes are respiring. There's drugs that have like quinolone regeneration effect, beta-lactams, there's phylogenetically distributed resistance. But there are two classes, macrolides and tetracyclines, that seem to target every microbe that we tested in this initial screen. And we went further and we saw that actually this kind of broad spectrum that those drugs have and their ability to kill some microbes and just inhibit others is what was causing the large impact they have on microbiome composition. But what I want to show you here is that even if you know that those drugs have a large impact, a collateral damage on the gut microbiome, can you do anything to ameliorate this effect or mitigate it? And we have this idea that, antibiotics are broad spectrum for a reason, and they're good in targeting many different pathogens, but as a collateral damage, they might be targeting also a lot of other microbes. So one idea is to come with antibiotics that they're very specific to the pathogen, but nobody is developing new antibiotics, so developing more narrow spectrum antibiotic is not something that the pharma would go. But maybe one idea would be to make those available antibiotics, make them more narrow spectrum. And the idea is that you could make them more narrow spectrum by adding a second drug. And we came with this idea because we knew that although a lot of drugs are acting as single drugs the same across many different species, the way they interact is very species-specific, and that came out from a large study we had done on antibacterial drug combinations on many different pathogens. So we thought that we could find a drug that basically will mask the effect of the antibiotic on the commensal, but still allow it to work on pathogens. So we took some of the most prevalent abundant species in the gut. We knew that tetracyclines and macrolides completely kill them. So we wanted actually to find strong antagonistic interactions with tetracycline and macrolides. We did that for a couple of different bacteroidid species. We screened the same library that I showed you before, 1,200 different drugs. We found 10 strong antagonisms. You can see here the data. Those are basically the antagonistic scores. those antagonists work in a number of different commensal species, and conveniently, they don't affect the antibiotic effect on the pathogen. This is what I'm showing you here in this plot. It's a bit complex, but I'll walk you through. So what you see on the y-axis is basically the ability of microbes to grow. We put one of those macrolides, erythromycin, and we do that as a function of those second drugs that we have. We call them antidotes because they can mask the effect of erythromycin on commensals. So every trace you see here, you see basically all the gray and blue traces are commensal species, all the red, purple traces are pathogenic species that macrolides are used for. So you can see how erythromycin inhibits as a function of addition of decumarol. So if you just put erythromycin alone, it inhibits everybody. As you start adding decumarol, then you see that a lot of the gut microbes can start growing, whereas the pathogens are completely inhibited. So this works in isolation. It actually works also if you assemble communities of the same species. And it also works in ex vivo personalized communities, so if you take communities from stool of individuals or even in animal models. So the take-home message from this one is that you can actually, antibiotic effects are there, and they have a well-known collateral damage in the gut microbiome, but I think there's a strong dependence on the class, and there's many more studies that they needed in that, and it's the antibiotics with a broader spectrum and the ability to selectively kill some of the members of the microbiomes that they might be the most dangerous for long-term microbiome composition and changes. And you can start thinking about how to mitigate some of this collateral damage of the antibiotics by using combinations that will allow the antibiotic to still act on the pathogen but will mask its effect on commensals. Now, the third part that I want to show you is that till now I've been showing you our ability to test drugs on single species, but those species are not living alone. They're living as part of communities in the gut. So we want to see whether our ability to know how each of those species in the community interacts with the drug allows us to predict what will happen in the community. And that's work, again, done with Per Borg's lab and Michi Zimmermans and Kira Patil, so spearheaded by Sarella Garcia, an ex-postdoc in the lab that has just now moved, and Michael Kuhn from Per Borg's lab. So this is the simple principle. So you know how a drug will interact with a single species. Now you put a complex community and you ask whether you know how the drug or how the single species will interact as the context of the community. And if everybody behaves the same in isolation or as in community, if you just measure the fitness in isolation and the fitness in the community, there should be a fairly good correlation. So this is what we did. So we had 30 different drugs in 3 different concentrations, and we have a synthetic community of 32 different species that we know it's stable and you can stably propagate it for many generations. So we knew, as I told you, how each of those species behave. And the question was that whether community response is the sum of the individual or the emergent behaviors. So here I'm showing you one of those drugs, so this methotrexate. I told you that here I'm plotting on the y-axis the community fitness, so how each of those individuals behaves in the community in the presence of the drug versus how it behaves alone in the presence of the drug. And if they behave the same, then you should have a fairly good correlation. And then you have those off the diagonal parts that you have protection, so things that are growing well in the community and normally would not grow well. And here is where you have sensitization. So here you have things that would normally grow well, but they don't grow in the community. So you can do that exercise in many different drugs, and here's the results. Just to make the point that you see basically emergent behaviors across every single drug. You see much more cross-protection, so they're common, but they're not the majority. And here you can see that for every drug treatment, even the different concentrations, before I was showing you just one concentration, there's emergent behaviors, but they're the minority. In most cases, you can predict what happens, but there's quite a bit of cross-protection and cross-sensitization. Now, we had different concentrations of the drugs, which allow us to ask the question whether those emergent behaviors change as a function of how strong the stress is. And that's what we're doing here. So we have the low drug concentration, the middle drug concentration, and the high. And we ask how much cross-protection we see. And what we see is that as the drug concentration increases, actually the ability of the community to protect individual decreases. And not only the ability of the community to protect individual decreases, but also the sensitization, so things that normally are not sensitive to the drug but become sensitive to the presence of the community, increases with increasing drug concentration, which actually tells us that there's a lot of community stability because of this prevalent cross-protection. So species normally that would be sensitive alone are protected by other members of the community, but this community stability actually collapses when the perturbation is getting stronger. The cross-protection mechanisms are not working anymore, and you see much more of those antagonistic interactions. So what I quickly showed you here is that most species actually respond the same to the drug in community as they respond in isolation, but there are emergent behaviors, and about a quarter of the emergent behaviors we see on those drugs that we tested. The most common emergent behavior is protection, so species are protected in the context of the community, but what we see, this ability to protect species dissipates at high drug concentrations, and actually a lot of negative interactions start emerging. So I would say that community resilience and stability, even in any microbial ecosystems, would have those tipping points that they can withstand environmental pressures and environmental harsh conditions, but up to a certain point, and then you start losing the ability to protect individuals. I didn't show you data, but we know the mechanisms that are behind this cross-protection, and we can go down to the species and enzyme-specific mechanisms that allow for cross-protection. And this knowledge allows us actually also to use that knowledge to modulate specifically communities. So I think I'll stop here. I said all the people who did the work in the lab, I thank funding agencies and lots of collaborators. Thanks for your time. Happy to take any questions.